It can get a little dry at times, so you're going to have to stick with me, but we'll talk about how a clinical trial is designed and performed more from the family, the patient and family perspective. Although I have to say, I've been involved in clinical trials for a while. I actually learned stuff putting this talk together, and it comes down to 5 steps to bring sort of a new therapeutic to market. We'll talk about sort of the current state of therapies that target genes, and I'm not kidding when I say this, but I think this is probably one of the most exciting times to be involved in medicine, because we're finally getting to the point where gene therapy is starting to pay off. It's almost unbelievable. And then we'll talk about therapeutic development for rare diseases and some of the things. How do rare diseases kind of fit in the overall setting of bringing a drug to market? So here are the steps. There's always steps involved. So these are the 5 steps in drug development that we'll go through. I put this slide up more just so there's a lot of online information if you're interested, and NORD is probably one of the best resources, especially if you want to become involved. The first step is sort of this idea of discovery and development, and it's probably the biggest sort of step in bringing a drug to market, and it's the hardest sort of to define, because it depends on where the idea for the drug came from. A lot of times a company or an academic will have sort of new insights into a disease process and say, hey, I want to target this pathway. Recently, with machine learning and having robotics, we're able to test a whole bunch of compounds if we know it's going to work. Some of the existing treatments that have unanticipated side effects, like Viagra, are ways that some drugs get to market. And then there's a lot of new technologies, and we'll spend some time talking about genetics. At this stage of development, there can be 1,000, 1,000,000 compounds, and there can be a lot of hope. But you have to realize getting from this stage all the way through to get a drug approved is difficult. It's very difficult. So I just threw up an example for discovery and development. And there on the graph there, I didn't label it, but I'll show you, that on the top there is everybody's normal cholesterol, where you fall. When you go to see your doctor and he or she looks to see where your cholesterol falls, you can see where you would be on a normal curve. You can see the graph below. Don't we all wish we were on that graph below, where that population of patients, their normal cholesterol is low. And it turns out these patients have a mutation which actually benefits health. They have an inability to produce LDL, and so they have very low cholesterols, and they go about their life not having to worry about the stuff that I have to worry about. Take a baby aspirin and a Lipitor. And it turned out that when they were looking at cholesterol metabolism, they looked at this group of families who had low cholesterol, did genetic testing on them. By doing genetic testing, found out, hey, they have a mutation in a gene called PCSK9. And someone said, well, can't we make everybody into a PCSK9 mutant? What happens if we take that approach? And so that's an idea of discovery and development, that someone spends a lot of time looking at LDL metabolism or cholesterol metabolism and says, maybe we should target this pathway. So that's one example of this idea of discovery and development. The next step is basically to say, hey, I have a drug. Is it going to work? Can I make it? Can I manufacture it? And you really have to start with experiments in the lab. And sometimes you'll hear the term in vitro, which means it's sort of outside the body. And then there's some in vivo experiments. Usually those are animal studies. So that is where we take a whole bunch of compounds, or let's say I want to modify a gene. You might start with animal models. So that's the preclinical research. So that's all the stuff that goes on before a drug is actually said, hey, we're going to try it in humans. The next step is the one probably people are most interested in and want to participate in, and this is this idea of clinical research. That is, I have a compound, I have a therapy, and I want to see if it works. I want to see if it's safe and effective. And that, you're going to hear that term over and over again. Safe and effective. And the researchers really have to come together and agree on a trial to say, in the trial you'll hear a lot of times it's the protocol. how do we test this drug? And that is really, believe it or not, what makes or breaks a drug. Because if you don't spend enough time in designing the trial, it won't be successful. And a lot of that has to do with how many patients you're able to enroll. And so you can see some of the things that go into trial design. Who qualifies to participate, how many people will be part of the study, how long will the study last, whether there'll be a control group, meaning there's some patients who get the treatment and some people don't get the treatment. What are you looking at? What's your measure of success? And how the data will be reviewed and analyzed. And this can be very difficult because sometimes for some diseases, and I'll just say, hey, I'm a nephrologist, the FDA wants what we call hard endpoints. So in adults, they want to say, hey, you have a new treatment for end-stage renal disease? I want to know how many people you save from dialysis, how many people you save from dying, right? Well, if you want to do a trial in children, right? Children, for whatever, because they're healthier, we can't design a trial that's going to look at mortality, right? That trial would take 30 years. So then we have to go to the FDA, hey, what are you willing to use as a surrogate or an alternative endpoint? So it's very tricky when it comes to trial design. So many a great treatment has been dropped at this point, trying to develop a trial design. Now, as far as the clinical research, the terms you're going to hear are basically the various phases of clinical research that can happen. Phase 1 is easy. Phase 1 is we take a healthy group of volunteers and give them the treatment and see what happens. So these are people not affected by disease. And it's really just a quick look at safety and dosage. So that's the very beginning. And you can see approximately 70% of drugs, because they've been tested in animals and in vitro, make it to the next phase. The next stage is phase 2. Phase 2 is really, does the treatment work? Is it effective? And phase 2 starts with a smaller group of patients. Here I have sort of 300 to 3,000 volunteers. You have to realize if it's a rare disease, you might not have 300 volunteers who all have the disease. So sometimes the clinical trial is modified based on how rare the disease is. You can see here about a quarter of the drugs then make it through to the next phase. And then the sort of the final phase is where it's kind of a go, no-go, that the FDA is going to look at the data, is phase 3. Often they build upon phase 2 and say, hey, we're going to look at a large group of patients and we're going to look at efficacy and side effects. And you can see now we're down to a quarter. We're sort of slowly chopping away at drugs, make it through this. So there's a couple of reminders I have to give to you. One is bolded there, right, is that it's a rare disease, right? You're not going to have a lot of participants. The FDA is aware of this. The flip side of that, and this is why it's always so amazing to come to a conference like this, is that the power of advocacy, the power of a small group. If you look at the treatments that are approved in cystinosis, the reason those treatments are approved is because people took part in clinical trials. That is, they said, hey, I'm willing to enter my child in a clinical trial. And that shows you the power of how powerful a small group can be to bring a treatment to market, even though it's an extremely rare disease. The final thing I have to remind you is that, when the FDA and trial design, children are part of a protected category for a lot of reasons. Not just because they're cute and we love them, but because they don't necessarily understand what the trial involves and the risks, right? And they usually have an advocate or a surrogate. So children fall into a group of protected classes. So are prisoners, so are protected women. So designing a trial for children is an extra hurdle as opposed to the rest of us who can consent. So a lot of times, that's an extra kind of barrier that you have to get through. If you're going to design a trial for children, there's some specific circumstances. Okay, so just graphically, which a lot of people like seeing, is here you see the phases of trials, basically. Remember, phase 1 is just healthy volunteers. Phase 2, you're really starting to look, is the treatment going to work? And phase 3 is that sort of that proof of concept. Not all drugs follow this. If the phase 2 results are so great and there's no side effects, occasionally the FDA will give early approval, especially if there's not another treatment out there or if it's a life-saving treatment. But this is the general idea when you hear about drug development and how drugs move through. And if you're interested in becoming part of research, if you're a healthy volunteer, you can participate in phase 1, but often the people affected are going to be recruited for phase 2 and phase 3. Does that make sense? That was kind of the dry portion of the talk, going through study design and all that other stuff. I will tell you that FDA approval, this is stuff I think if you asked everybody, the experts in the audience, this is still stuff that is a little bit of a mysterious part of how a drug gets approved. Because it's a very involved process. And there's no question about it, the FDA is a very conservative organization, right? They want to prevent ineffective and dangerous products reaching the market, right? And so they are very cautious, judicious when it comes to approving a drug. And basically the drug manufacturer has to take all the data, the preclinical data, the clinical data, put it in a package and hand it off to the FDA. The FDA then goes through a process. It's usually, you can see, 6 to 9 months to say, hey, do we approve this drug? And that can mean that, hey, you're assigned the preclinical data, the group of you are assigned the clinical data, and then they all get together as a group. Occasionally, and you'll hear about this in the news, especially with new treatments for Alzheimer's, if there's some controversy about the treatment, the FDA will actually get a committee of experts together, and they all end up voting on a treatment. And the FDA has a balance. They're trying to come up with the idea that the treatment is efficacious, means it works, and it's safe, right? So if you're just going to say, hey, Dr. Zaritsky, I come to market, I want to treat sunburn, right? Well, in most cases, sunburn isn't you're going to get better for sunburn. You better believe that the FDA is going to look very, very carefully at the safety data, right? Because the risk versus benefit in treating sunburn is much different than you're treating a life-threatening disease. So even if you look at the latest drug approved for Alzheimer's, there's some serious side effects of that drug. There's some serious side effects like cerebral edema, but the FDA has still given its approval because they recognize how devastating the disease it is. So the risk and benefit is really what goes into that. And sometimes that's, that can be a very difficult decision to make, a very difficult balance. And there's people out there who says, well, I don't care what the side effects are, I'm willing to take the treatment. When we get to stuff like, getting rid of my wrinkles here or something like that, you better believe for the FDA to approve it, they're going to say there really has to be very few side effects to make that balance. Okay. Finally, I'll just say that once the drug has been shown to be safe and effective, there's a label. So if you ever want to know anything about the drug you're taking, you can read what the FDA labeled. That's sort of the summary of all the data that's gone into getting that drug approved. Okay? I'll make mention is that once a drug is approved for a specific indication, clinicians, caregivers can use that drug, what we call off-label, right? Meaning that we use it for a different disease, right? And we as clinicians are kind of weighing that balance between safety and side effects. So that's that big difference between on-label, where someone, it's been studied, and this is the package insert, versus off-label. As parents of children with cystinosis, you have to recognize that a lot of the drugs that we use to treat children are off-label, right? Because there hasn't been studies in children, and we've kind of assumed that what's happened in an adult is going to happen in children. Does that make sense? So you'll hear a lot of times your doc talking about label versus off-label. Oh, wait. Well, we still do have some more dry stuff, so we're almost there. Step 5 is sort of the post-marketing. So occasionally a treatment makes it through, and then people realize, hey, there's some side effects that were not noticed. Because when you look at a study, maybe you've only studied 1,000 people, but 100,000 people have been exposed to the drug, you'll see some side effects. So there might actually be another trial in that period that can cause some phase 4 trials after the drug's been approved. But this is this idea where really the true safety of the product evolves over time. Sometimes actually the FDA will rescind its approval based on post-marketing adverse effects. Oh, and here's sort of the summary of bringing a drug to market. So you can see, on average, of the 5,000 potential treatments that enter preclinical testing, only 5 make it to testing in humans, and only 1 is approved. So that is, the odds are stacked against you bringing a treatment. And then it takes an average of 12 years to get from an experimental drug from preclinical research all the way through approval. So although we often get very excited about a new treatment being out there, it might be on the nightly news, it often takes a very long time for that treatment to make it through the FDA and actually get approved. And then you can see here about 14% of drugs entering clinical trials will be approved. And then this is there's a little bit of controversy, is how much does it actually cost to get a drug approved? And you have to realize some of that cost is built into the preclinical work, paying for scientists, paying for this drug development, paying for research. But it's an extremely expensive and long process. Oh, the exciting stuff. Maybe it's not that exciting, but I find this stuff really exciting. So, and some of this, for you, some people in the audience, you're gonna say, I don't need to hear about genes anymore. But some of you haven't heard it. So just gonna start a little basic, and this idea of what is a gene. So if we look within the very building block of your body, there's cells. Cells make up organs. Organs make up your body. And within that cell, there's a nucleus, which is sort of the control center of the cell. Within the nucleus, there's a set of instructions that we're all born with, which is chromosomes. You can now know that it's 23andMe, you know how many chromosomes you have, and all this stuff. And within the chromosome, there's DNA. And within the DNA, there's a gene. The way I typically explain it is, think of your genes as a set of blueprints that go to building the house, that's you, and maintaining that house. All of us are given 2 sets of blueprints. And when we look, sometimes there's errors in those blueprints. In some cases, you can still build the house with 1 correct set. So it says, hey, one set of blueprints says, hey, put the toilet bowl upside down. It's not going to work like that. But the other set puts it in the normal. So the body reads the correct way. Sometimes you need both sets of blueprints to make a successful gene product. And the big thing is, what we're learning is, as we understand the genome better and better, not all errors in blueprints will result in changes on the building of the house. And so this is something that we're all individuals. We all have slightly different genetic coding that makes us up. Not every change results in what we would call, an error, right? So we're still learning. But the beautiful thing about this understanding is, now we are getting to the point where we're actually able to take those blueprints and modify them. Sometimes we're able to sneak in an extra set of blueprints that says, hey, make sure you put the toilet the right side up. Or sometimes we're actually able to go in and physically fix the blueprints and get the correct genetic information in there. So that was your introduction to genetics on a Friday morning. So, and then the idea is, we've heard about gene therapy forever. It's the wave of the future, what we're going to do. And this is really the idea that where we are now, we are standing on the shoulders of giants. If you look at the work that's gone into gene therapy, and we've had some bumps on the way. One of the first gene therapy products out there actually caused a death, and that actually slowed the whole process down. But where we are now is, we're on the part of the curve that we're going to see a whole bunch of new gene therapies. And diseases that we thought we'd never be able to treat are now treatable. And some of these treatments, I will say, are actually cures. So if you look at some diseases like sickle cell, there's a potential that there's genetic therapy, or hemophilia, a genetic therapy was just approved. That's actually a cure for the patient. It's not even a treatment, because they've been able to fix the blueprint. So here's this idea of the explosion that we're seeing, that if you go way back when, into the early 90s, there was just a handful of potential gene therapies. And this only goes up to 2019, but the curve has really become astronomical. And the people leading the way are really those patients treating leukemias and lymphomas, those bloodborne disorders. And almost some of the gene therapy is becoming, I would say, it's starting to push to be standard of care, as opposed to experimental or last phase. And it's becoming sort of part of our nomenclature, really, you know. So, like I mentioned, gene therapy aims to fix that problem in the blueprint. And this is where we can get into a lot of the details. You don't necessarily have to know the details to understand gene therapy. You just have to be aware that gene therapy includes several different tools when you think of those blueprints that you're given. One of the things you're going to hear about in a little bit is this idea of adding a gene. So let's say your blueprint is missing how to install the garage door in your house, or you're missing a transporter in your lysosome, right, as in cystinosis. Some of the therapies now have the potential to really sneak in an extra set of blueprints, a modified version that the nucleus then uses to produce the correct product. And I'll show you that we've really been sneaky about ways of getting that extra instructions into the cell. And we've actually taken viruses that were not good for you and able to kind of genetically engineer them so that we use them to deliver what we want. You hear a lot about CRISPR and other things of gene editing. This is this idea of using a molecular scissors to say, hey, we're going to cut out this bad part of the blueprint and put in a new part. And then really what's really taken off is this idea of gene silencing. And this is the idea, as we start to understand genes better, we realize it's not just the standard that we all learned in high school, that there's a gene, the gene is read and translated into a protein. We realize that there's a lot of control. One of the controls are little pieces of genetic material called RNA, which can actually silence genes. So for example, right now, there's several treatments that target the liver and some of the things the liver do. If we could just turn the liver off from making cholesterol, right? If we could turn the liver off from making something that causes hypertension, we can give you one shot a quarter or one shot a year that builds up in your liver and silences that gene product. So this stuff is just unbelievable. It's like science fiction. So some diseases that I've treated, rare diseases, one of them called primary hyperoxaluria, it's now being treated by one of these RNA silencing techniques. So a disease that, you know, we used to have to do a kidney and liver transplant to treat the patient now is being treated by a single injection every 3 months. And then for me taking my Lipitor, right, and eventually getting hypertension, I'm waiting for that to happen as well, I'll get a shot once a year. So it's just unbelievable where we are and the explosion of therapy. So I'm just going to show you this because the rock star who's coming on after me is going to mention one of these viruses, these viral targeting vectors. So a virus is a very unusual thing in where it's a small package of DNA, and or DNA or RNA. Its job is just to replicate itself, so it's able to invade a cell and sneak in extra instructions, right? So it basically takes over your house and tells it to make whatever it wants, and it usually destroys the house at the end of the day. What very smart people have been able to do is say, hey, well, let's take that and modify it so it doesn't destroy the house, but it tells the house to make something additional. And this is this idea of a viral vector or a vector that we use to introduce gene therapy. There's two different forms of vectors. One of them just inserts an extra copy. The other, the lentivirus, which you'll hear about, actually goes in and actually adds the gene to your set of genes. But this is one of the amazing things about gene therapies. How do you get genes in there? We've been able to take things that were bad for you, viruses, and actually able to change them so they do what we want them to do. I think that's incredible. So one type of gene therapy which you're going to hear about coming right after me is this idea of hematic stem cell therapy, right? And basically what this is basically a gene therapy where we take out your bone marrow cells, take them out of the body, modify them, and put them back into your body. Probably what you guys have heard about in the press is something called CAR T therapy. Who's heard of CAR T therapy here? Yeah. This is a therapy that's it's basically been for people with terminal cancers who have not been able to, go into remission for whatever reason. That used to be a death sentence. CAR T therapy is a novel therapy that really has revolutionized the care of cancer, where they take a bunch of your T cells out and use a viral vector to modify those cells. So they're able to recognize cancer cells, they grow those cancer cells up and throw it back in your body. And it's when you read about this stuff, you're like, when I went to medical school, you learned about, well, this was the survival rate of this type of cancer, this type of cancer. Things have really turned on their head. And the evolution has been, it was just approved in 2017. Now it's starting to become pushed forward to not quite first-line therapy, but certainly second-line therapy. So it's really here and now. What's so cool about the hematic stem cells is that these cells, like your blood cells, they can go out and deliver products to the rest of your body. And this is I'll show you this on the next slide here. This is a process called cross-correction. So, for example, in the talk you're going to hear about, we're actually able to take a disease that isn't really necessarily a disease of white blood cells or red blood cells or macrophages, but we're able to then give those cells instructions how to build something. Those cells then go out and donate their product. So, for example, if you have let's say your kidney cell has a faulty lysosome, right? It's actually possible to develop, to modify your own stem cells, to give them instructions how to make a good lysosome, put those stem cells back inside you, then those stem cells go out and actually donate a functioning lysosome to some of the other products. So HSC has been used primarily for treating leukemias and lymphomas, with the idea is those new cells go out and attack and get rid of the cancer, but those new cells can also go out and donate stuff. And so that's what you're going to hear about in very shortly. And here's this idea. Basically, they take your stem cells out, and we're actually able to isolate stem cells. They modify them genetically, grow them up, and then put them back in the body. And then those stem cells are yours. They belong to you. They're able to replicate themselves and go out and develop, either fight whatever they need to fight, produce whatever they need to produce, or sometimes donate basically gene products or proteins to other cells that you haven't genetically modified. It's a very quick, and in some ways, I don't have to fix the whole body, right? I just fix the bone marrow, and then the bone marrow goes out and fixes the rest of the body. So incredible therapy. So I need to now move into rare disease considerations, and I just want to, the big point of my talk here is that what gene therapy offers is that we're starting to blur the lines between the treatment of a rare disease and a common disease, because the techniques that we're using are almost interchangeable depending on what disease. So I'll tell you, like this company that makes the silencing RNA therapy. I was talking with them because I'm very interested in kidney disease, and they said, Dr. Zaritsky, that's very interesting, but do you know any other liver diseases that we can, where the liver makes another protein? So literally, they want to treat something as common as hypertension, but if you have an extremely rare liver disease, they just use the same technology. They use the same identical technology, so they can mass produce sort of genetic treatments, whether you have an extremely common liver disease, like fatty liver disease, or a disease that's 1 in a million, like oxalosis. That's the excitement and the promise of gene therapy, right? Okay, I'm almost out of time. So just some rare disease considerations. We have to recognize that pharmaceutical companies, they're commercial enterprises. Sometimes it's very difficult to convince a pharmaceutical company to develop a treatment for a rare disease because it's going to be a small market. It can sometimes be very difficult to develop a treatment for diseases that affect patients of lower economic status, such as like tuberculosis. So most of traditional drug development has been targeted to a first world with very common problems. Cholesterol, Lipitor, right? The funny thing is, when you guys have heard this over and over again, rare diseases are common. Right? Look, 25 to 30 million Americans are living with a rare disease. That's like 1 in 12, 1 in 11. And of the 7,000 known diseases, fewer than 10% have a treatment, and even fewer have a cure. So what the beauty of the Orphan Drug Act in 1983 was basically a bipartisan effort to say, hey, we need to recognize that rare diseases are more common, and we need to spend some attention to it. So I don't have all the time, but of this Orphan Drug Act, it sort of opened up the ability and the efforts and opened the eyes of the drug companies, sometimes in good ways, sometimes in bad ways, to say, hey, let's start developing drugs for rare diseases. And you can see the explosion in the number of drugs that have developed since the Orphan Drug Act. Sometimes we wonder, the government, it doesn't do a lot. It's, there's not a lot of progress. They're just raising taxes. And this is an example, I think, that most of us in this room would say the Orphan Drug Act was an example of bipartisan legislature that has moved the needle, that has actually brought the interests and to a rare group of diseases. So there's some other considerations here. This I throw up here because the Orphan Drug Act doesn't come without its critics, because you could say, well, did a drug company just take an existing treatment and ask for a grant or remarket it? You can see here that I can't remember what year this is, like 2015, the top 10 selling drugs, the one in blues are actually orphan drugs, which is amazing. But what is further amazing is that a drug that's developed for a rare disease, like if we look at rituximab that was developed to treat a B-cell leukemia, that drug now has so many off-label treatments for both common and rare diseases. So this is this idea again that if you develop a treatment for a rare disease, you might be able to apply it to multiple diseases. So the treatment you're going to hear about, using the lentivirus to treat cystinosis, that has the ability to treat many, many more rare diseases down the road. So just to summarize, because I'm running out of time, that road, you'll hear it from bench, the preclinical stuff, where someone sits with a pipette or does something to a mouse, from bedside is very long and expensive. I want to really, you got to take, the cystinosis group has been an amazing example of advocacy, where they've been able to take and move drugs from the preclinical stage all the way through to approval, even though there's not that many people to study. So that is a huge, that's a great way to move the needle. Genetic therapy represents a revolutionary step in treating human disease. I think that idea that, I'm only going to develop something for rare disease or something for common disease, that's being very blurred. Remember, this drug company said, hey, Dr. Zaritsky, what liver diseases do you know? They didn't necessarily, they already have a vector. They know how to silence genes in the livers. They just want, hey, what diseases can we treat now? Which is amazing. There still is a question on how expensive these treatments will be. So, for example, if you look at the genetic therapy, the RNA silencing therapy for that PCSK9, the guy who developed the therapy said, I want this to be a treatment that everybody in the world gets, that we're able to lower everybody's cholesterol, right? Well, they have to somehow pay for the development, and you see that treatment's about $10,000 a year. So you still have to deal with the expense of the treatments. And then finally, I'll say it for the 100th time, what's unique about gene therapy is that a treatment developed for a rare disease can be rapidly adopted to treat more common diseases, and vice versa. So I think that it really offers a potential. And if I look at, well, I'm getting older now. You can see the boot on my foot. And I mentioned I'm taking Lipitor and all that stuff. But some diseases I thought would never have a treatment are now being treated. And I feel myself like the old fogey when I have a resident and they're like, oh, just give a dose of rituximab. I'm like, back when I was a resident, we didn't have that fancy drug. We had a, so that's what's so amazing. So diseases that people have lived their whole life with, there's just new treatments now, which is amazing. Oh, hey, that was the opening act, so get ready.